Hi, this is Alistair Ewing. Again, welcome to the next video interview in our series to introduce new and exciting companies and technologies to the life science and medical device markets. Today, the company I'm going to introduce to you is a company called Fida Bio. They are based in Denmark, and this is a company that's developed a novel system essentially to help you quantitate and characterize proteins and do analysis a little bit different from anything else that's on the market. So I'll be talking with Brian Sorensen, who's the CEO, and Henrik Jensen, who is the Chief Scientific Officer. Okay, so let's get going. Okay, welcome Brian and Henrik, and thanks for taking time to talk to me today about Fida Bio. So could you tell me a little bit about the company and your main products?
Yeah, I can talk about the company.
Yeah.
At least initially here, the company is a spin-off from University of Copenhagen. The technology we're working with is actually a technology that researchers from the university has been working with for 10 plus years. And about 5 years ago, probably, we took over the patents and used 3 years or so to develop an instrument, something we did in the spare time. And then 1.5 years ago, we concluded that now we had a product that was viable, and we also could see that there was an interest in the market of actually working with it. So about a year ago, we decided that it was too much to do on Sundays and Saturdays, and we went out to get some funding. And as of August last year, we became full-time with the company, and we started also engaging other people, and now we are a small organization. And that seems to be going quite well. Even though there's a lot of technologies there seems to be a room for our technology also. But we can get back to that. The technology, Henrik?
Yeah, so the technology is it's called flow-induced dispersion analysis, and it's a flow-based method where we use a microfluidic flow capillary. And because it's a microfluidic flow system, it means that it consumes very small amounts of sample in the order of nanoliters to microliters. We can measure directly in very complicated samples, such as, for example, plasma and serum. Mm-hmm. The technology is actually based on an old principle called Taylor dispersion analysis, and this is something we combined with ligand-based measurements. So we now have a method where we very sensitively can measure molecular interactions under in crude samples in nano to microliters sample amounts. The instrument that we are marketing now is a fully automated instrument where you can load vials or 96 flow plates, so 296 flow plates at a time. And it's walk-away automation, so you simply place your samples and you let the instrument do the measurement for you.
Okay. So why did you develop this product? What were the sort of market drivers that encouraged you to develop the product?
Well, we felt that there was something missing in this segment. So I had been working at the university before I joined the company here, and often we were using surface-based procedures for measuring molecular interactions, and we realized that often they fell short. There were a lot of artifacts when you put, for example, a protein on the surface. It can actually change quite a lot, and what you measure on surface-bound proteins is not necessarily how they behave in vivo. So with a colleague, I developed the technology, and we patented at the University of Copenhagen. And then, of course, we did a market search, and it turned out that we were not the only one having problems measuring molecular interactions in crude samples. And I guess it's safe to say that these drawbacks of surface-based procedures is widely recognized in the field. And this is also what we can see now from the first customers using the instrument. I think they find it's quite easy to use and reliable. what also turned out was that the advantage of being able to measure on very small amounts of sample, that has actually enabled a lot of new projects to be initiated at our customers, because a lot of the current methods were actually requiring quite a lot of sample material. So now you can do full projects with just a few micrograms of expensive protein. So that we see is something that seems to be an advantage at many customers.
So
and maybe also with respect to the technology, it's basically determination of size or size changes that occur when you have a binding event, and as it's well known, a lot of biologic properties are actually linked to size and changes in size. So by then, it's a way of getting not just normal KDs or concentration measurements, but also more complicated analysis of the molecules that you're working with, or the nanoparticles you're working with.
So the benefits to a user who's working on protein analysis, protein research and characterization, for example, is the small volumes, speed of analysis, and the fact that you're not there's no matrix inhibitors or very few matrix inhibitors. Is my understanding?
That's correct. And as Brian also hinted to, we also find that actually the additional information that you get from the absolute size measurement is also key to some projects. Simply, it gives new insight. So of course there are some operational advantages, but there are also some more how to say, specific scientific advantages that seem to be valued highly by our customers.
So I would maybe add a fourth bullet point to the ones you're mentioning. The fourth bullet point being that you can work with quite large sizes. Also, we can work with molecules up to 1,000 nanometer in diameters, which are quite few technologies that are capable of doing that.
What about specific applications? Are there any applications where your potential customers are very focused on, that they want to talk more about compared to other applications?
Yeah, I think these applications we have where we're using small amounts of sample and measuring directly in complicated sample matrices. That's something that has been of interest to our customers from the very beginning. And I guess more recently, what we see is that projects where you have multiple bindings, where you have formation of molecular constructs in solutions, oligomerization, and so on, there we really see the benefit of being able to measure the absolute size, because then we can simply say whether it's one or two protein that binds and for example forms a dimer or tetramer or hexamer, that we can see directly from the measurement. And I think we are quite unique there compared to other technologies. The same way we can see if we have antibody binding, does one antibody bind, or is it two antibodies, and so on. So these are readouts that you don't get in any other technologies.
So if we look at the specific applications that our current customers are running, then it is, as Henrik has already alluded to, it's defining or confirming the oligomeric state of the molecules that you are working with. It's finding complicated interactions, bindings between, for instance, peptides and liposomes. It's quantification of antibodies directly in crude media. It's working with exosomes. So a lot of the things that you could maybe define as modalities that are at the forefront of the current drug development. Very recently, we have been looking quite a bit on degradation, and it also seems that the technology lends itself beautifully to characterizing some of the key components of the new degraded drugs, PROTACs, and what they're called.
Right. Exactly. Most of your customers or your target market is biopharma and the pharma industry, essentially.
Biopharma as well as academia.
academia as well. Okay.
Yeah, it's.
Yes.
I mean, we're still working with. Let's not pretend anything. We're working with a small customer group, but it's more or less 50-50 between biotech, industry in general, as well as academia.
Can I ask what might be a stupid question? This is a label-free method, isn't it? So, or is it not? How do you detect your?
We detect using a fluorescent readout, and in our standard configuration, we label one of the binding partners
Okay.
with the dye. We also have the possibility of doing intrinsic fluorescence. So many proteins contains tryptophan or tyrosines, which fluoresce in the UV regions, and in those cases, it's completely label free, of course.
Okay. And you provide all of the consumables and reagents, or can people source them from, you know, another
Yes.
company?
Yeah, I mean, we provide the basic reagents such as the microfluidic flow channel that is used, and also consumables such as the special vials and the 96 well plates that goes into the instrument is something that we provide. And of course, the customer then comes with the specific system that they wish to investigate using our platform.
What technologies that exist today would you hope to displace with this? You know, obviously people are doing certain interactions and
It's, of course, there are technologies out there that we would like to displace, Alistair, but the way we see this is actually our target audience are those who have yet not entered into any marriage with existing technologies. Those who have existing technologies, that's, I think, a trait of the market that they very much like to work with their technologies. Also, when they suddenly are standing with challenges that are not directly in the sweet spot for the technologies, and they do whatever they can to stretch their current technologies to also cover that area. So where we see an opening is two ways: those who have not yet married a specific technology, and of course also we can see that there are more and more situations where the existing technologies simply cannot address the challenges that the companies or the researchers are sitting with. Most recently, we've seen somebody working with specifically sticky proteins, which they have been struggling with for a long period of time, and they have really tried to take their current technologies and see if they could find ways of compensating for the stickiness of the protein, and had to give up. And in that situation, it's nice to see that they can take advantage of our in-solution technology and that it works well with them. Okay. So we're not to sum it up, we're not, our main strategy is not to knock somebody off. It's to get a foothold with the questions that cannot be answered with the current technologies.
Okay. And what are your, you know, the company's relatively young. What are your main marketing channels? How are you selling this?
It's direct sales, right? And it's distributors. And until recently, it had to be in a singular form, distributor, but now it can be in a plural form because we have just signed an agreement with another distributor. So it's early days. You can say the secondary market, if somebody wants to be called a secondary market, it's through distributors, and then the direct markets is something we try to address ourselves directly. And it's not because there are many good ideas, there are many good reasons for working with distributors, but of course, as being a young company, having a technology that we know a lot of, it's still so that getting the feedback directly from the early adapters is key to us, and that's the reason why we have said that in the main markets we would like to go direct.
So if you look at the marketing and in terms of pricing, my understanding is you complement other technologies, and your technology helps people do things today which they can't do with existing technologies. But you know, you're always competing for funding. How would you look at your product in terms of pricing? Is it competitive with the complementary products, or are you?
Yeah, we've been looking at what complementary products, how they are priced, and we have placed ourselves in a sweet spot there, so that it's approximately the same price as the comparative products comparative products that do not have auto samplers. We are more in the same price range, but with an auto sampler. And then I also think that in terms of consumables, we are the low end. And if you will add in labor cost, it is really it's set up, work away. A lot of the other technologies require that there's a scientist, PhD, constantly overviewing what goes on. That's not the case here. You can leave it to a skilled technician, and they place the samples in the instrument, and it's literally walk away. So overall, we're competitive in many different ways without going out and offering a bargain.
Right. As you know, these are short interviews, so just a couple of final questions. One is, for anybody watching this, what important benefits would you like to highlight that have not come up previously in our short discussion?
And it's for my part, and then you can supplement it. It's probably that, and that's what we hear from customers, that it's very easy to use. You very fast you become comfortable with the technology, and it's not unusual that we install the instrument, we leave them in the afternoon, and the next day they say, you know what, we already got the first results that we like, and we actually have a lot of confidence in it. So for my part, it would be ease of setting up new assays.
Yeah, I agree. And I mean, we had, for example, one customer who had one instrument, and then they requested actually a second instrument because there were so many people using it. So it's very common that that users start using it quite intensively just after a few days. We find that it's very easy to install students in universities in using the equipment, and they are rapidly get familiar with the technology. We also took a lot of effort in the software development to make it user friendly, and we have, for example, an assay development tool where you can generate your assay parameters. You get a list of the exact solution that you need to prepare and how to prepare them and so on. So it's not only the measurement it does, it's actually also telling you how to do all the practical stuff that's always surrounding these kind of assay in the laboratory. And we find that has been quite popular actually among our customers.
So to customers out there, if you're sitting with anything that you're struggling with in terms of characterization, functional characterization, then try it out, and you will find out when you start to use it on the things that you cannot do with other instrumentation, that is actually also useful for all the ordinary stuff.
So, but it's mainly proteins, or are there other applications that you can highlight benefits for?
So we have a lot of we have quite a number of applications recently on particulate systems. So, for example, binding to nanoparticle, binding to liposomes. We got a paper out recently in collaboration with the University of Copenhagen. We have some projects in the pipeline where we're looking at virus particles, for example, and so on. And these are measurements that are quite difficult to do with other technologies, and also where we see the added benefit of being able to measure on tiny amounts of samples seems to be of high benefit for our customers.
And I don't know if maybe you already mentioned it, Henrik, but elsewhere it's also important that we literally can detect directly in crude samples. It can be in plasma, in serum, in cell lysates, and fermentation meters, CHO cells, whatever you're working with. You don't need any sample preparation. You may need to dilute it a little bit, but there's no sample preparation as such that is needed.
Okay. Well, I had a look at your website. There's a lot of applications information. There's a good video with you, Henrik, showing or describing the technology. So for anybody that wants to find out more, where do they go to get this information?
Well, our homepage is certainly one good place to go, and we have a section on the homepage saying applications, where you find a lot of application notes and also linked to scientific papers where we and others have used the technology.
Yeah, or elsewhere the phone is actually a quite efficient way of getting more information.
And the web address is fidabio.com, is it?
That's right.
That's right, yes.
FIDABIO.com.
That's right.
Exactly.
Yeah. You can also find us on LinkedIn and social media and so on.
Okay, great. Then thank you very much. Let's leave it there. And I really appreciate your time. Thanks, I enjoyed it.
Thank you to you, Alistair.
Thank you.
Thank you.
Thank you, Alistair.
Bye.
Bye.